Welcome to this step-by-step guide on PLGA nanoparticle synthesis. In modern drug delivery, scientists are constantly searching for ways to deliver medicines more precisely and safely inside the human body. One of the most powerful tools developed for this purpose is PLGA nanoparticles. Tiny biodegradable carriers capable of protecting drugs, controlling their release, and targeting specific tissues. In this presentation, we will explore how these nanoparticles are synthesized in the laboratory, why different synthesis methods exist, and what practical considerations researchers must keep in mind while designing effective nanoformulations. The goal is not only to understand the process, but also to gain insights from real-world formulation experience. PLGA, or polylactic coglycolic acid, has an interesting history. It was first developed in the 1970s for biodegradable surgical sutures, such as Dexon and Vicryl. Researchers later realized that this polymer could also serve as an excellent carrier for controlled drug delivery, leading to its widespread use in pharmaceutical nanotechnology during the 1980s and beyond. One of the biggest advantages of PLGA is its biodegradability. The polymer breaks down through hydrolysis of its ester bonds into lactic acid and glycolic acid. These molecules naturally enter the body's metabolic pathways and are eventually converted into carbon dioxide and water through the Krebs cycle. This makes PLGA extremely safe and suitable for medical applications. Several techniques exist for synthesizing PLGA nanoparticles, and choosing the right one depends primarily on the drug's solubility and the desired particle size. Each method has unique advantages and is optimized for specific types of molecules. For example, the single emulsion method works well for hydrophobic drugs, while double emulsion is better suited for hydrophilic compounds like proteins or DNA. Nanoprecipitation is known for its simplicity and ability to produce very small nanoparticles, and microfluidics provides highly controlled and reproducible particle synthesis, especially useful in clinical manufacturing. The single emulsion technique is one of the most commonly used methods for encapsulating hydrophobic drugs. The process begins by dissolving PLGA and the drug in an organic solvent, such as dichloromethane or chloroform. This organic phase is then introduced into an aqueous solution containing a stabilizing surfactant like polyvinyl alcohol. Through sonication or high-speed homogenization, the organic phase breaks into tiny droplets suspended in water. As the solvent evaporates during continuous stirring, these droplets harden and form solid PLGA nanoparticles. Controlling the emulsification energy and polymer concentration is critical, as these parameters strongly influence the final particle size. When dealing with hydrophilic molecules such as proteins, peptides, or nucleic acids, the double emulsion technique becomes more effective. This process begins by emulsifying an aqueous drug solution into an organic PLGA solution, forming the first water-in-oil emulsion. This primary emulsion is then dispersed into a second aqueous phase to form a water-in-oil-in-water system. As the solvent evaporates, the polymer solidifies around the inner aqueous droplets, effectively trapping the hydrophilic drug within the nanoparticle structure. High-speed centrifugation is typically used afterward to collect and purify the nanoparticles. Nanoprecipitation, also known as solvent displacement, is a simple and efficient technique widely used to produce small PLGA nanoparticles. In this method, PLGA and the drug are dissolved in a water-miscible organic solvent, such as acetone. The organic solution is then slowly added to an aqueous phase under stirring. As the solvent diffuses into the water, the polymer rapidly loses solubility and precipitates into nanoparticles almost instantly. This rapid formation process often leads to particles with narrow size distribution and is particularly useful for hydrophobic drugs. Microfluidic synthesis represents a more advanced approach to nanoparticle production. Instead of mixing solutions in bulk containers, microfluidic devices use tiny channels where organic and aqueous phases meet under precisely controlled flow conditions. By adjusting parameters such as flow rate ratio and total flow rate, researchers can control nanoparticle size and uniformity with remarkable precision. Microfluidics also offers excellent reproducibility and scalability, making it highly attractive for pharmaceutical manufacturing and clinical applications. After nanoparticles are formed, their surfaces can be modified to improve biological performance. One common strategy is pigylation, where polyethylene glycol chains are attached to the nanoparticle surface to create a stealth effect that helps avoid immune detection and prolong circulation time. Another approach is ligand conjugation, where molecules such as antibodies or peptides are attached to enable targeted delivery to specific cells, such as cancer cells. Surface charge modification is also important, as it influences cellular uptake, biodistribution, and interaction with biological membranes. During nanoparticle synthesis, several practical challenges may arise. For example, particles larger than 300 nanometers often result from insufficient mixing energy or excessive polymer concentration. Increasing sonication power or reducing polymer concentration can help address this issue. High polydispersity, which indicates uneven particle sizes, is usually caused by poor mixing conditions. In such cases, improving stirring efficiency or switching to microfluidic synthesis can significantly improve uniformity. Drug leakage and nanoparticle aggregation are also common issues that can be addressed by optimizing formulation conditions and stabilizing agents. Scaling nanoparticle production from small laboratory batches to industrial manufacturing requires careful process optimization. Techniques like microfluidics and tangential flow filtration are often used to maintain consistency when producing larger quantities. In addition, pharmaceutical production must comply with good manufacturing practice standards. This means validating solvents, surfactants, and analytical methods, while also ensuring sterility through aseptic processing or terminal sterilization techniques, such as gamma irradiation. PLGA technology has already led to several successful pharmaceutical products. For instance, Lupron Depot uses PLGA microspheres to provide sustained drug release for prostate cancer treatment. Another example is Neutropin Depot, which delivers growth hormone through a controlled release PLGA system. Looking toward the future, researchers are developing PLGA-based systems for advanced therapies such as RNA delivery and gene editing technologies like CRISPR. Artificial intelligence is also being used to optimize nanoparticle formulations, accelerating the design of next-generation nanomedicines. Developing effective nanoparticle formulations often requires specialized analytical techniques and formulation expertise. Laboratories with advanced capabilities can help researchers characterize particle size, stability, drug loading, and release profiles to ensure successful product development. With the rapid growth of nanomedicine, collaboration between formulation scientists, analytical experts, and pharmaceutical developers is becoming increasingly important for translating innovative nanoparticle technologies from the lab to real clinical applications.